Hello friends, welcome to our YouTube channel, The Exploring Medico. Today we'll learn something about erythropoiesis. In this topic we'll cover the definition, then site of erythropoiesis and various stages of erythropoiesis. So we'll start with the definition. Erythropoiesis, the word itself suggests that erythro means for erythrocyte and poiesis means production and development. So the definition is like this: erythropoiesis is the process of the origin, development, and maturation of erythrocytes. Then hemopoiesis. Hemopoiesis is similar to erythropoiesis. It is the process of origin, development, and maturation of all the blood cells. Hemo means all the blood cells, and poiesis means its production and development. Now we'll see the site of erythropoiesis. In fetal life, erythropoiesis occurs in three stages. The first one is megaloblastic stage. It is seen during the first 2 months of intrauterine life, means 2-month pregnancy. The RBCs are produced from mesenchyme of yolk sac. Then second one is hepatic stage. From the 3rd month of intrauterine life, and liver is the main organ that produces RBCs. From the 3rd month of intrauterine life, the hepatic stage starts, and the liver is the main organ that produces RBCs. Spleen and lymphoid organs are also involved in erythropoiesis. They have minor role. They don't play the major role in the production of erythropoiesis, but yes, some of the role is there. Then third stage is myeloid stage. During the last 3 months of intrauterine life, from 7th to 9th months of intrauterine life, this myeloid stage is there. In this stage, the RBCs are produced from red bone marrow and liver. So we have seen 3 stages in erythropoiesis. We'll revise it. The first one is megaloblastic stage, then second is hepatic stage, and third one is myeloid stage. We have to remember these 3 stages and the sites where they are produced during these 3 stages. Now in newborn babies, children, and adults. So up to the age of 20 years, RBCs are produced from the red bone marrow of all the bones, that is long bones and flat bones. During puberty age up till 20 years, RBCs are produced from red bone marrow of all the bones. And after the age of 20 years, that during the age of maturation, RBCs are produced from membranous bones like vertebra, sternum, ribs, scapula, iliac bones, and skull bones, and from the end of long bones. Only these bones and the specific areas of long bones have red bone marrow cells which produces RBCs. Only membranous bones produces RBCs after the age of 20 years. Now we'll see the process of erythropoiesis and its different stages. So first is committed PHSC. These cells are restricted to give rise to one group of blood cells, and the name suggests that they are committed. PHSC means only one group of blood cells is formed from specific PHSC, that is committed PHSC, and they are of two types, that is lymphoid stem cells and colony forming blastocytes. We'll see one by one. Lymphoid stem cells, It gives rise to lymphocytes and natural killer cells. So, lymphoid stem cells, as the name suggests, first it will give rise to lymphocytes and then it will give rise to natural killer cells. And the second one, that is colony forming blastocytes, gives rise to myeloid cells. Myeloid cells are the blood cells other than lymphocytes. Then myeloid cells, when grow in cultures, these cells form colonies, hence the name new colony forming blastocytes. So these myeloid cells form different colonies, and so they are also named as colony forming blastocytes. Now, the stem cells. Stem cells are the primary cells capable of self renewal and differentiating into specialized cells. So, these stem cells are the base of the process of erythropoiesis. From that cells only, the various different blood cells are formed. They are the primary cells and they are capable of self renewal. They get renewed by self and differentiating into specialized cells. They differentiate into various different specialized cells. Hemopoietic stem cells are the primitive cells in the bone marrow which give rise to blood cells. So these hemopoietic stem cells, they are the most primitive cells in the bone marrow and they give rise to these blood cells. Hemopoietic stem cells in the bone marrow are called uncommitted pluripotent hemopoietic stem cells. As in earlier slide we have seen PHSC. So this is they are these hemopoietic stem cells are also called uncommitted pluripotent hemopoietic stem cells, uncommitted PHSC. Earlier we have seen committed PHSC, which gives rise to a specific group of blood cells, and these hemopoietic stem cells are uncommitted PHSC. So they don't have the specific characteristic to give rise to only one group of cells. PHSC is defined as a cell that can give rise to all types of blood cells. As I have said earlier, they are the cell that can give rise to all type of blood cells, not a specific group of cells. Now, different units of colony forming cells are, there are different units in colony forming cells. First is CFUE, means colony forming unit erythrocytes. Cells of this unit develop into erythrocytes. The next one is colony forming unit granulocytes or monocytes, CFUGM. These cells give rise to granulocytes, and these granulocytes, granulocytes that are neutrophils, basophils, eosinophils, etc., and monocytes. The third one is colony forming unit megakaryocytes, that is CFUM, and they give rise to platelets. From these, platelets are developed. So there are different units of colony forming cell. We have seen three: CFUE, CFUGM, and CFUM. First is for erythrocyte, second is for granulocytes or monocytes, and the third one is for megakaryocytes. Now we'll see different stages of colony forming unit E cells, erythrocyte cells, and matured RBCs. So there are 6 stages, total 6 stages are there. We have to remember the order, it is very important for us. And the stages and process of erythropoiesis is one of the IMP questions in the blood portion. So 6 stages of erythropoiesis are: first one is proerythroblast, then early erythroblast, then intermediate normoblast, then late normoblast. Fifth one is reticulocyte and then matured erythrocyte. Total 6 are there. We'll discuss it one by one. So the first stage is proerythroblast and it is also known as megaloblast. It is the first cells derived from CFUE colony forming unit erythrocyte. So it is the first stage of a process of erythropoiesis. This cell it is very large in size with a diameter of about 20 microns. Its size is very large. Its nucleus is large and occupies the cell almost completely. So it has a very big nucleus which almost occupies the whole cell completely. The nucleus has two or more nuclei and a reticular network. So this nucleus have two or more nuclei in it and has reticular network. Proerythroblast does not contain hemoglobin. So it is the main characteristic in which it does not contain hemoglobin. Hemoglobin appears during the last two stages of erythropoiesis. So in this there is no hemoglobin present. The cytoplasm is basophilic in nature, basic nature. Then proerythroblast multiplies several times and finally forms the cell of next stage. It multiplies several times. Now the second stage is early normoblast. This cell is smaller than proerythroblast with a diameter of about 15 microns. So this is smaller than proerythroblast. Proerythroblast was 20 microns while this is about 15 microns in diameter. In the nucleus, the nucleoli disappear. So it does not have nuclei. Nuclei is not visible in the cell. Then condensation of chromatin network occurs. Here the condensation of chromatin network occurs. The condensed network becomes dense. The cytoplasm is basophilic in nature. So cytoplasm is basophilic while the condensed network becomes dense. It becomes thick and increased in volume. It becomes dense. Hence, this cell is also called as basophilic erythroblast. So, early normoblast is also called basophilic erythroblast due to its basophilic nature. This cells develops into next stage called intermediate normoblast. So, this we have seen early normoblast, and now we'll see intermediate normoblast. So, this cell develops into the next stage, that is intermediate normoblast. Intermediate normoblast. This cell is smaller than the early normoblast with a diameter of 10 to 12 microns. So as we move on to the maturation stage, the diameter of the cell decreases. So in this stage, the diameter of the cell becomes 10 to 12 microns, which is lesser than the early normoblast. The nucleus is still present, but the chromatin network shows further condensation. So nucleus is present, but chromatin network shows further condensation. The hemoglobin starts appearing. So as I have said, in starting there is no hemoglobin present in the cells. While we move on to the maturation stage of erythrocyte, the hemoglobin appears, and the starting of appearance of hemoglobin is from here, that is intermediate normoblast. So cytoplasm is already basophilic as in earlier stages. The cell develops into next stage called the late normoblast. Uh, Earlier we have seen early normoblast. This stage is intermediate normoblast, and from these cells it will develop into late normoblast. So now we'll see in late normoblast stage. The diameter of the cell decreases further to 8 to 19 microns. 8 to 9 microns, not 19, it is a mistake. 8 to 9 microns. It is the diameter is very small compared to the first stage, that was 20 microns, and in this it is 8 to 9 microns only. The nucleus becomes very small with a very much condensed chromatin network, and it is known as ink spot nucleus. So nucleus becomes very small, which can not be seen sometime. So it is also known as ink spot nucleus. The quantity of hemoglobin increases. In earlier stage, the hemoglobin has appeared, which was in a very small amount, and now the quantity of hemoglobin increases. Cytoplasm becomes almost acidophilic, so the cell is called as orthochromic erythroblast. Here the cytoplasm becomes the acidophilic, earlier it was basophilic. And due to this acidophilic nature of cytoplasm, it is also called as orthochromic erythroblast. In the final stage of late normoblast, just before it passes to next stages, the nucleus disintegrates and disappears. And the process by which nucleus disappears is called pycnosis. So the disappearance of nucleus is called pycnosis, and the nucleus also disintegrates and disappears. And this disappearance of nucleus is seen in the final stage of late normoblast, the stage where late normoblast is about to develop into the next stages. Then the stage before the maturation of erythrocyte is reticulocyte. It is otherwise known as immature RBC. So reticulocyte is also known as immature RBC. It is slightly larger than matured RBC, slightly larger, not much. The cytoplasm contains the reticular network, and that is why also it might be called reticulocyte. Due to the presence of reticular network, it is called as reticulocyte. As I have said, there is much reticular network, so it is called reticulocyte. Here the cytoplasm will be acidophilic, as we have seen in earlier stage. Now we'll talk about matured erythrocyte, that is red blood cells. Reticular network disappears and the cell becomes the matured RBC and attains the biconcave shape. In this stage, the reticular network disappears, it totally disappears, and the cell becomes matured RBC. So it becomes mature and attains the biconcave shape. The cell decreases in size to 7.2 microns in diameter. It becomes of the smallest size, that is 7.2 microns in diameter, as compared to the earlier stages. The matured RBC is with hemoglobin but without nucleus. So this is the main point, that matured RBC is full of hemoglobin but it does not have nucleus. The nucleus disappears totally. It requires 7 days for the development and maturation of RBC from proerythroblast. From the first stage to the last stage, that is from proerythroblast to mature RBC, it requires 7 days for the development and maturation. It requires 5 days up to the stage of reticulocyte. Reticulocyte takes 2 more days to become the mature RBC, as we have seen. So total 7 days to complete from proerythroblast to mature RBC, and from reticulocyte to mature RBC it takes 2 days. So this is the chart. The first is from up we'll see to all. That is, first cell is pluripotent hemopoietic stem cell. That is the most primitive cell. From that they are subdivided into two. That is colony forming blastocyte and lymphoid stem cells. Lymphoid stem cells further divides into lymphoblast, and then it forms lymphocyte. While colony forming blastocytes, they have 3 types of colony forming units, that is CFUE, CFUM, and CFUGM. That is E for erythrocyte, M for monocytes, GM for granulocytes or monocytes, M for megakaryocyte. That second one is megakaryocyte. So CFUE, it is the process of forming the erythrocyte. So from CFUE it will become to proerythroblast, then early normoblast, then intermediate normoblast, then late normoblast, then reticulocyte, and finally the erythrocyte. This colony forming unit megakaryocyte will be divided into megakaryocyte, and then it will develop into platelets. Then colony forming unit granulocytes or monocytes. It has 2 types, that is myeloblast and monoblast. Monoblast will develop into pre-monocyte, and then it will develop finally into monocyte. Myeloblast is developed into pre-myelocyte, and it has 3 types of it is subdivided into 3 types of cells, that is neutrophil myelocyte, eosinophil myelocyte, and basophil myelocyte. As the name suggests, they will finally develop into 3 types of cells, that is neutrophil, eosinophil, and basophil. From myelocyte it develops into metamyelocyte, and then it will become mature neutrophil, eosinophil, or basophil. Okay, so this is the whole chart of development and production of various blood cells. This is a very important chart, and in exam if we write this chart, then it will cover most of the marks. So it is very important to remember various stages of erythropoiesis. This table is very helpful for that. In proerythroblast, the main event is synthesis of hemoglobin starts. In early normoblast, nucleoli disappear. It is the important event in this stage. Then in intermediate normoblast, hemoglobin starts appearing. In proerythroblast, synthesis of hemoglobin has started, but it was not seen, it has not appeared. But in the intermediate normoblastic stage, hemoglobin starts appearing. In late normoblast, nucleus disappears in late normoblast stage. Then in reticulocyte, reticulum is formed and cell enters capillary from site of production. In matured RBC, reticulum disappears and cell attains biconcavity. So RBC becomes mature and reticulum disappears and cell attains biconcavity. So this is the end of our topic of erythropoiesis. So in case of any doubts, you can ask in comment section, and please like the video and subscribe our channel. Thank you.